Hello everyone, welcome to another video from the drug delivery lab. We just had another paper accepted and I have the lead author Vishal, PhD student in my lab with me, and we will just discuss the paper to give you a perspective from our side. So Vishal, how does it feel?
It feels great. I am very relieved to say the least, and I'm very happy that our work is now will be now read by a lot of people in the field, and we'll receive a lot of inputs on it.
Great, yeah. So can you briefly describe this paper?
So this paper is a way. So the basis of this paper was to generate a new in vitro culture system for understanding host mycobacteria interaction. So the current culture systems you are mostly 2D surfaces. So basically, cells, immune cells are differentiated on 2D surfaces and are infected with mycobacteria. And this is the main in vitro culture system that is being used by researchers globally for tuberculosis research. So we thought that if we go in with our expertise of hydrogels and uh, design a better in vitro culture system, wherein we use collagen 1 as the main component of the hydrogel, because collagen 1 is the main component of the human lung, where mycobacterial tuberculosis infection manifests. So we thought that if we use collagen hydrogels, monocytes, and mycobacteria, and see if that becomes a better culture system for carrying out some basic in vitro research in tuberculosis.
Cool, yeah, and we have also done a lot of matching with the lung young modelers and even
We have also
the pore
looked at
size.
the pore sizes, then we have looked at the phenotypic and genotypic similarity of the pathogen and as well as the host with human infections in general. So a lot of studies were carried out in that aspect. And also we use this study to evaluate if the first line of antibiotics are able to clear the mycobacteria or not. So and this forms the basis of the paper, and this how the paper goes.
Cool. And so what are the unique aspects of this? Why does a person working on TB should use this model?
So this is because this we believe this is one of the first culture systems wherein we have used monocytes and collagen hydrogels, and we see that we see very interesting results. For example, we see the formation of foamy macrophages in our system. We see the formation of mycobacterial cords in the system. All these are phenotypes which are relevant to human infection, and they can be studied much better using one single platform as such. And then we also see a very interesting result in the form of pyrazinamide, which is a first-line anti-tuberculosis drug. So it happens that pyrazinamide was a chance discovery. It was discovered directly on animals, and now we see that using our culture system, we see pyrazinate's efficacy at human relevant concentrations. So that gives us a basis of how we can study of how pyrazine can be effective or is effective in killing mycobacteria. And it's a first line drug, so it's a very interesting compound drug to be looked at.
Yeah, so essentially, yeah, as Vishal is saying, what we see is that this culture system represents a very good testing system because, as seen by pyrazinamide, no other in vitro model works at the clinically relevant concentrations. Plus, we've seen a lot of genotypes or phenotypes that are matching in our system much more closely with the human system as opposed to current in vitro setups. So, okay, so what are the future implications of such a model now?
So, as I was mentioning briefly about pyrazinamide, the immediate goals can be that you understand how this antibiotic is actually working in killing the mycobacteria in the human cells. So that would lead us to, so if we can figure out the mechanism of action, we can understand or we can discover new class of antibiotics altogether. And a more interesting model that we are currently working on is generating using the systems to now generate human tuberculosis granulomas, which are being characterized And so it has a lot of promise in that aspect.
Great. So a little bit moving away from the science. How has been your experience working on this project for now several years? How would you describe that?
Yeah, so this I am a mechanical engineer by training, and the lab gave me and Rachit gave me the opportunity to basically work on a biological problem, which I consider myself very lucky to be working on. And it has been a great learning experience to merge different fields in such way, like use my understanding of mechanical concepts and other things to see if we can directly uh, generate in vitro platforms which can further our understanding of human health and tuberculosis infection in this particular case. When I started off, I never imagined that the project would help me in developing so many skills altogether and giving me fresh perspectives on how human health and disease should be looked at. So it's been a very enriching experience in that.
Okay, thanks, Vishal, for those nice words, and I hope you guys enjoyed this version of the author's take. Please do check out this paper in Advanced Healthcare Materials. I'll attach the link in the description, and see you all next time. Bye.
Bye.